the last thing we talked about was alcohol fermentation. Right? Remember, for fermentation, this is the process of glycolysis that's actually linked to another set of steps, chemical reactions that help us to regenerate NAD+, so they can go back and do glycolysis again and again and again. Remember, for alcohol fermentation, this was a two-step process. We took our pyruvate, 3-carbon molecule, we removed one of the carbons that left as carbon dioxide, and then what remained was a 2-carbon molecule called acetaldehyde, and this functioned as an electron acceptor, so here's our acetaldehyde, it accepted our electrons from NADH. Effectively, it helped to oxidize NADH back to NAD+, and by picking up those electrons, acetaldehyde became that ethanol. Alcohol fermentation, 2 steps in addition to glycolysis. Lactic acid fermentation, the other kind of fermentation that we're going to be talking about in class, is a little bit more simple. In this case, you only have 1 step in this process. So you still have glycolysis, right? Same deal as before in alcohol fermentation. You take glucose, you send it through glycolysis, you end up producing pyruvate. In the process, you manage to make a little bit of ATP, and you also reduced NAD+ to NADH. Again, NADH needs a place to drop off those electrons when oxygen is not present, because it can't drop them off at the mitochondria to go into oxidative phosphorylation. So instead, for lactic acid fermentation, super easy. The pyruvate itself, the end product of glycolysis, will feed back, pick up the electrons from NADH. It effectively helps to oxidize NADH back to NAD+, by picking up those 2 electrons, and pyruvate thus becomes our lactate, or lactic acid. Now, some fungi perform lactic acid fermentation, some bacteria do this. These are used to make cheeses and yogurts in terms of our food making industry. Now, you also have cells that can do fermentation. Your human muscle cells can undergo fermentation. And this happens when, let's say, you start exercising, right? You're working out, you're doing great. Right at the beginning, you're not huffing and puffing yet, right? Your breathing is normal, until all of a sudden, your muscle cells are like, whoa, hang on, we've used up all of our ATP. We need to make more. So it tries to pick up its production of cellular respiration, and it uses up all the available oxygen. And for a brief period of time after you've started working out, your muscles are without oxygen. They've used it all up to try to make more ATP to keep you going. And so for a brief period of time, your muscle cells say, I can't breathe anymore, I don't have any more oxygen, I can't do cellular respiration. Well, your muscle cells don't die, right? They don't cease working because they run out of ATP. No, they switch. They switch from doing cellular respiration in the presence of oxygen to doing lactic acid fermentation in the absence of oxygen. And so they take the pyruvate, they use it to pick up those extra electrons from NADH, and they produce lactate, or lactic acid. And your muscle cells continue to do this, making a little bit of ATP, allowing you to keep going, until all of a sudden now you're huffing and puffing, and your breathing rate, right, increases to accommodate that need for oxygen. Oxygen starts to flow back to your muscles, and now your muscle cells can go back to doing the much more efficient cellular respiration. Okay, so there are different kinds of cells. For example, those that are obligate anaerobes. These are going to either always carry out fermentation or anaerobic respiration, and these guys usually can't survive in the presence of oxygen. So they have to make their ATP either using anaerobic respiration or fermentation without using oxygen. There are also those cells that are obligate aerobes, or they can only do aerobic respiration. They don't have any other options. Your brain cells, the nerves, the neurons within your brain, these cells are obligate aerobes. They need oxygen. If you cut off oxygen supply to your brain, those cells can't switch to fermentation. Their only way to make ATP is by using oxygen in cellular respiration. This is why it's so important to maintain that oxygen supply to your brain. Because if they don't have it, they will die, unlike your muscle cells. Your muscle cells, and then yeast and many other bacteria, for example, are considered facultative anaerobes, meaning that they can survive using either fermentation or cellular respiration, aka aerobic respiration. For a facultative anaerobe, right, essentially they'll do glycolysis just like everybody else to produce pyruvate. And then at pyruvate here, this is a fork in the road. If oxygen is present, the pyruvate will be shuttled into the mitochondria for the remaining 2 steps of cellular respiration, making a whole bunch of ATP. On the other hand, if the cell goes through glycolysis, produces the pyruvate, and oxygen is not available, then it'll switch, and it'll start either doing alcohol fermentation or lactic acid fermentation, for example, just to survive until perhaps oxygen supply resumes, and then they can switch back to doing cellular respiration. So that pyruvate is essentially that fork in the road. Oxygen is present, it goes to the mitochondria. Oxygen is not present, and it's used as part of fermentation. It's a nice little backup mechanism or insurance mechanism for those cells. They're a little bit more flexible in terms of whether they can live under oxygenated or not oxygenated conditions. Okay, so let's do a little bit of an exercise. In this chart, you guys can fill in as we go along, but it's essentially a comparison between aerobic respiration, anaerobic respiration, and then fermentation in terms of use of glycolysis, the oxidizing agent for glucose, the process of oxidizing NADH, and then the amount of ATP produced. So let's go through it step by step. Number 1, do these 3 processes use glycolysis? To help you complete this, remember that glycolysis does not depend on oxygen, and it doesn't depend on the mitochondria, so essentially anybody can do it, no matter their method of creating ATP. So in aerobic respiration, do we use glycolysis? Yes, we do. In anaerobic respiration, where that ending substance at the bottom of the electron transport chain is no longer oxygen, but is something else, are we going to use glycolysis for that first step? Yes, we will. And then lastly, in fermentation, do we use glycolysis in fermentation? Yes, we definitely do. Remember, that's the way that we make ATP. Glycolysis is the only way we make ATP, and the other half of fermentation is just a couple of extra reactions, 1 or 2, to regenerate that NAD+. That's it. And then we rely on that glycolysis to make that ATP. Okay, let's think about oxidizing agents. Remember, an oxidizing agent is one that accepts electrons from something else, and it effectively helps to oxidize or lose the electrons from that other substance, right? So what is the oxidizing agent during glycolysis? What helps to remove electrons from glucose? It's a hard one. You think about it. Remember, from glucose, that electron acceptor is going to be NAD+. It gets reduced to NADH in aerobic respiration. Anaerobic respiration, same process, right? We're only changing what's at the bottom of the electron transport chain. The rest is essentially the same. Okay, what about in fermentation? What oxidizes the glucose? Again, it's going to be NAD+. By accepting those electrons from glucose, it becomes NADH. Same all the way across the board. Okay, now, once we have the NADH, once the NADH has taken, or the NAD+ has taken the electrons from glucose, where does it give them up? Where in aerobic respiration do we give up those electrons from NADH? Well, remember, that's going to be the electron transport chain, right? In anaerobic respiration, same idea. The electrons get given up to that electron transport chain. Only difference is what's waiting down at the very bottom of the chain, waiting for those electrons at the bottom. What about in fermentation? Here's a tricky one. Where do the NADH molecules give up their electrons? Well, there is no electron transport chain in fermentation, right? Because the whole mitochondria has shut down. So where do those electrons go? Well, this is going to differ whether we're talking about lactic acid fermentation or alcohol fermentation. So either it's going to be the acetaldehyde, if we're talking about alcohol fermentation, right? That's going to grab those electrons from NADH, oxidizing it back to NAD+, Or in lactic acid fermentation, the pyruvate directly is going to accept those extra electrons from NADH, again, oxidizing it back to NAD+. All right, let's think about how many ATP are produced. For aerobic respiration, remember it's about 32 ATPs. For anaerobic respiration, it's a little bit less than what you have for aerobic respiration. And this makes sense if you think about it. Right, oxygen is one of the most electronegative atoms out there. If you're going to use something else at the bottom of the electron transport chain to pull those electrons down, it's probably going to end up not being as electronegative as the oxygen. So it won't be able to pull as much of that energy from the electrons out as the oxygen. And so the process won't be able to produce as much ATP as you would expect to get from aerobic respiration. Okay. And our last one, how much ATP is produced from fermentation? Remember, in fermentation, the only thing producing ATP is glycolysis. And how many ATPs do we produce in glycolysis? 2. And that's it, a measly little 2. So clearly, the most effective way to produce ATP is through the use of oxygen in aerobic respiration. Okay, now we're going to jump all the way back to the very beginning of chapter 9, right? Remember that glucose is not the only molecule we can use as a source of energy, right? We can use other carbohydrates, we can use fats, we can use proteins. It all depends on the system, where the system's at. So really, both glycolysis and the citric acid cycle that help to degrade that glucose molecule, these are major intersections to various catabolic and anabolic pathways. We can feed molecules in to glycolysis in the citric acid cycle at various parts, and we can also funnel molecules out. If the cell has plenty of ATP, maybe we want to use some of those molecules as building material instead of letting them complete the process and go on to form ATP. So let's take a look at where proteins, other carbohydrates, and fats can actually feed into cellular respiration. All right, so for proteins, these guys, right, made out of amino acids, you chop up the amino acids, you have to deaminate them, or take the amino groups away, and then they can funnel in, for example, here at the end of glycolysis near pyruvate. For carbohydrates, there's a wide range of carbohydrates that can be used, right? We've started with glucose because that was easy, but remember, glucose gets converted to fructose in one of the first steps of glycolysis. So if you have fructose, just shove it in there, not a problem. Now, fats are a little bit more complicated, right? Remember, fats have those two parts. They have the glycerol and then the 3 fatty acids. So the fats are essentially going to be taken apart. The glycerol can be modified and used or converted into glyceraldehyde 3-phosphate, or G3P, that molecule right in between the two phases of glycolysis, right? Energy investment versus energy payoff. And then the fatty acids. Think back to our discussion of the peroxisome. What does the peroxisome do? It takes those fatty acids and it chops them up into those 2 carbon chunks. Remember, this is called beta oxidation. Well, those 2 carbon chunks are very similar to that 2 carbon part of the acetyl-CoA molecule. In fact, each of those 2 carbon chunks in the fatty acids can go into cellular respiration as acetyl-CoA. And this is how those fatty acids and the fat molecules can store a ginormous amount of that energy. Because you're taking 2 carbon chunks at a time, and each one can just be directly fed into the middle of cellular respiration. Okay, and then lastly, the proteins, amino acids, can also be modified, not just to go into glycolysis, but also into pyruvate oxidation as well as the citric acid cycle, which makes them really very versatile. All right, and then everything, proteins, carbs, and fats, all their energy ends up being funneled into oxidative phosphorylation, and then it moves on to the production of ATP.